topics. As I told you, it is a multidisciplinary field. So broadly defining, in broader sense, there are 3 to 4 topics are most widely studied. The first topic, it is called as comparative genomics. So as the name says, comparative, genomic means comparison between different genomics here. So in this, what we do, we try to compare genomes such as mouse and human, that is chromosomes, or you try to compare the genes. So one genes or several sets of genes, and try to compare functional regions of the genomic regions here. So the comparative genomics has several applications here. So it can provide many insights from new information. So it can tell about gene locations, or you can find out genes which are present in a novel genome or newly sequenced genome, and you can find out transcription factor binding sites, and then you can construct phylogenetic trees. Phylogenetic trees means the relationship between different organisms in evolutionary context here. For example, in this case, if you see, so these are sequences, these colors are sequences for different proteins and from different organisms here. So copper, molybdenum, nickel, cobalt, selenium, iron and zinc, these are utilizing proteins and sequences downloaded from various organisms such as bacteria, archaea and eukarya. So simply comparing this protein sequence, you can tell their evolutionary context. So it is closer to the classical taxonomy. Archaea and eukarya are closer, and slightly bacteria is distant to these 2 classes here. Simplest sequence comparisons can provide greater insights, and then using comparative genomics we can do gene prediction, annotation of sequenced genomes, and perform evolutionary studies. You can study about protein families and SNPs, SNPs or single nucleotide polymorphisms. So all these things can be studied. So this forms the core of comparative genomics. And then coming to the next topic, it is called as structural genomics. As the name suggests, structural genomic. So trying to understand the structure of complete genome, which is organized in 3-dimensional space in nucleus located in 3D in a cell here. So it encompasses understanding the proteins which are wounded around or wrapped around the DNA sequence, such as nucleosomes or histone proteins, and the organization of DNA. DNA has several organizations: coding regions, non-coding regions, telomeric regions, introns, exons, promoters, cis-regulatory regions. So several types of DNA elements are present here. Basically the structural genomics is to understand the organization, the sequence organization, and also 3-dimensional organization of genome given in a organism here. So basically structural bioinformatics, its goals include here genome sequencing. That means genome sequencing. In genome sequence we have a step called as genome assembly. Sequencing is done through experimental methodology, and assembly is done through bioinformatics softwares here. So bioinformatics or structural genomics plays key role in assembling a unknown genome here, and the predictions of genes in newly sequenced genome. So there are several methods called as ab initio methods or comparative or phylogenetic footprinting methods to predict genes in the sequence genome here. So we can use bioinformatic algorithms such as hidden Markov models and neural networks, and you can also see the structure of the gene or location of a gene in a genome, find out introns, exons, splice sites, regulatory sites in a given or in a sequenced genome, and we can also compare gene maps of different organisms here. So basically structural genomics involves understanding the DNA in a cell at 3-dimensional as well as sequence level here. So, and there is a most important topic in bioinformatics, that is called as functional genomics. Basically, it is cataloging the activities of genes and gene interactions, and also interactions among various products here. So, the central dogma of biology says that DNA codes RNA, RNA codes protein. So, in comparative genomics, our major focus will be DNA, whereas in functional genomics, so our major focus will be the function of gene which is transcribed from the DNA to the translated protein. The overall function of a gene is called as, understanding is called as functional genomics here. So, in these things, so there are several topics are there: transcriptomics, metabolomics, epigenomics. So various topics will come here. So it can go to complete population level analysis or individual phenotypes analysis here. So it has several applications here. We'll see those applications in later classes here. So basically, the things which are majorly studied in functional genomics is study of gene expression using techniques such as microarray analysis, and sequence patterns, study of sequence patterns in promoter regions, and try to understand the overall transcription regulation through motif detection here. So along with this functional genomics, structural genomics and comparative genomics, there is another topic called as structural bioinformatics. Where basically we focus on 3-dimensional structures of proteins, RNA and DNA here. So that is most important aspect of bioinformatics. So the understanding the structure of a protein can be done through 3-dimensional structure. Designing drugs targeting the proteins which are involved in pathogenicity is done through structural bioinformatics here. So whatever